as a PhD student. So I converted my master's into PhD. So it essentially became like an integrated master's and PhD course. So Dr. Campbell's expertise was post-transcriptional regulation at that point of time. He was studying these RNA binding proteins, and he was studying these RNA binding proteins that bound to the 3' UTR of the RNA. If you don't know any of these terms, just ask me and I'll explain them. So the proteins that he was really interested in were called PUF proteins. So PUF proteins are post-transcriptional regulators, obviously, and they're highly conserved proteins. So they're present in all mammals, and they're present in worms, they're present in flies, and even plants, actually. So it's a very important protein to be studying. So they have a conserved role as well because they are conserved evolutionarily. So their conserved role is stem cell maintenance. And my project was studying a particular PUF protein in a particular model system. So what they do is they bind specific bases, really short bases, in the 3' UTR of mRNA, and they control the metabolism of mRNA. So essentially stability of mRNA. Consequently, they control gene expression, right? mRNA, protein, gene expression, etc. So in humans, there are 2 PUF proteins. They're called PUM1 and 2. I'll just say human 1 and 2, right? And their expression is really high in embryonic stem cells and hematopoietic stem cells and neural stem cells, etc. So anywhere where there's a necessity to regulate stem cell maintenance, you'll find PUF proteins. And so knockout of these genes, PUF genes, or absence of PUF function in humans is associated with developmental delay and fertility and neurogenesis and neural stem cell regeneration or renewal. And consequently, there are issues with learning, memory, etc. So you can already see that they play a very important role. So our pursuit was to elucidate some aspects of their function. And the model system that we chose was obviously C. elegans. So does anybody know what C. elegans is? If they do, then I'm not going to explain it. No?
No.
So essentially, hello? Anyone? Okay. So C. elegans are these small, tiny nematodes. They're hermaphrodites, which means that they express both sperm and oocytes. And they're also transparent, they're like smaller than 1 mm, tiny, microscopic almost. And you can visualize the internal, activity really well. So it's really an excellent model system to study several molecular mechanisms. And PUF proteins are highly expressed in the gonads of C. elegans, germline of C. elegans. So that was our focus. Our model system was C. elegans, and we studied the function of 2 PUF proteins in the C. elegans germline. So let me see where to start. In order for me to first use C. elegans as a model system, I had to train. So my professor actually sent me to Ithaca, and under Dr. Taven Lowe in Ithaca College, I learned how to, maintain C. elegans worms, propagate them, store them, and also, how to perform RNAi. And I also performed CRISPR with Dr. CRISPR-Cas, the mechanism. Okay, I'm going to skip that. So I also did that with her. So she essentially taught me how to work with C. elegans. And then I went back to UT Dallas, established C. elegans in my lab, and started my project. So my project was focused on two C. elegans proteins. One is called PUF8, another one is called FBF2. So PUF8 binds a 8 base element, and FBF2 binds a 9 base element. But even though they bind different length RNA elements, their structure is really similar. They're sort of C-shaped, and they have 8 structural repeats. So each structural repeat will define an RNA base that it will bind to. So we were really curious about what aspect of their structure allows them to bind to different length RNA and also perform different kinds of function. Because when you knock down PUF8, you get a tumor formation in the germline. And when you knock down FBF, you get a germline collapse. So they're opposite, but functionally, right? So we wanted to see if we could we wanted to see the relationship between motif divergence and biological function, difference in motif length selection and biological function. So what we did was we mutated FBF, which binds 9 bases, using yeast as a model system again, yeast-3 hybrid as a model system. And we selected a FBF mutant that binds to PUF8 elements. So now we have a mutant FBF2, which binds 8 bases. And what we did was we took our C. elegans worms and we knocked down PUF8 function using RNAi, and we observed what happened. So essentially, I already told you that PUF8 knockdown will cause a tumor formation, right? But we now have introduced an FBF mutant, which will bind to PUF targets. So it is supposed to function like PUF8. And we observed that it did. So when we introduced FBF2 mutant into the worms which did not have PUF8 function, we saw rescue of tumors. So it was actually, the pictures are really good. If you can go see my paper, I'm really proud of them. So if you go see my paper, you can see the rescue of tumor. It was pretty remarkable. And so let's see. So essentially, what this told us was that we can re-engineer the proteins, these proteins, to bind different elements and consequently change biological function. We also understood the relationship between, the length of the motif that they select and biological function. So we also collaborated with a crystal structure lab, and we generated a crystal structure of PUF8. And my first paper has all of this. So hopefully this adds to the plethora of information about PUF proteins, and we understand that family better with this. So that's all I'm going to talk about with respect to my project. But if you have any questions, please ask away. I mean, I'd rather interact with you guys, Hello? Rashmina?
Yeah.
Okay.
Students, if you have any questions on the research that she has explained, please ask your questions.
Ma'am, what is puff protein, ma'am?
Sorry?
You mentioned something about puff protein, no, ma'am?
Could you repeat that? I'm sorry, I can't.
He is asking what is puff protein? What is the puff protein is all about?
Oh, okay. So PUF proteins are RNA binding proteins. They bind to the 3' UTR of the RNA, and then they control the stability of RNA and its metabolism, essentially. So, which means that they control gene expression of that particular, you know, gene that's producing the mRNA.
Do you have any slides or some pictures of this Vandita with you?
Yeah, I do. Let me try and project this onto the screen. Okay. So let's see.
You have a
Present.
present now option there.
Yeah, I can see that. Wow. So can everybody see this?
Yeah, it is visible. It will be nice if you can show the UTR of that RNA and all, so that where it binds, if you have
Okay.
them also.
Oh, okay, I see. So this is the puff protein, if you can see my cursor. Okay, so when I spoke about the 8 structural repeats, this is what I'm talking about, you know. These are the structural repeats. And each repeat defines the RNA it binds to, RNA base it binds to. So this right here are the RNA bases. This is just a short RNA base in a long transcript. So we're only representing the short region in a long RNA. So they interact, you know, these repeats, they're called trims. They interact with the bases by edge-on and stacking interaction. So they essentially clamp onto a base. So that's how they recognize a motif. Is that clear? I'm not
Yes,
sure.
ma'am.
Yeah. So you know how. Let me just stop for a second. So you learn this in your biology classes, that in a transcript 3' UTR is like a bed. It attracts so many RNA binding proteins. They regulate the most RNA binding proteins regulate the transcripts using 3' UTR as their targets. So that's puff protein is one of those proteins.
Vandita, I have a doubt
Yeah,
or rather a question which I want to understand.
yeah.
So, you mentioned puff proteins are highly expressed in the early development, right? So, embryonic cells.
They're highly expressed in early development, but they also function later on as well in some
But
adults.
uh, is there a variation in the expression of these genes in the early development and in the later stages? They are overexpressed in the early stages and they are less expressed, or they're expressing in a controlled way in the later stages.
Yes, during early development, with C. elegans as a model system, the expression of puff proteins is very high compared to an adult, worm. So, because traditionally they are required for, stem cell maintenance, which is essential for development.
Yeah,
So,
yeah.
yeah.
So then are these puff proteins are also the part of the human homeotic genes or homeobox?
Yes, they are. They are actually. I wrote a whole section on that. Yes, they are, Rashmi ma'am.
Yeah. So that's something which is very interesting because there
Yeah,
is a lot of hope that is associated with this homeotic genes
I know,
with
I know.
respect to.
It was these are very early work, right? That's how it's the history of puff proteins. Homeotic genes were how puff proteins were discovered. They knocked down some genes and The Drosophila, fruit fly, didn't develop a head, and they realized
Yeah.
one of this is, yeah, you're right, it is.
Questions from students?
Yeah, you don't have to ask me about science. You can ask me about, what you want to do later. That's fine too.
Come on, guys, it's an interactive session. You can ask any basic questions that you are scared to ask your teachers or any of us. You can ask those basic questions to Vandita and get to know what is
Ma'am,
UR
16 is RN.
and
Hello, ma'am.
So, who wants to do what? Does anybody have an idea? Does anybody?
Sanjay has a question, Vandita. Sanjay is asking something. Yeah,
Oh,
Sanjay.
okay. Hi, Sanjay.
Hi, ma'am. Can this 16S RN be used to increase any enzyme production like that, ma'am?
What?
16S RNA, can it be used for enhancing enzyme production, ma'am?
Used for enzyme production?
Enhancing enzyme production is his question. 16 is rRNAs, which are in prokaryotic system, in fact. So his question is, can these 16 is rRNAs be used to enhance some enzyme expression or enzyme enhancement?
Enzyme and enhancement. Is it like?
Actually, it is not possible. 16 is rRNAs, which are the ribosomal RNAs in prokaryotic
Oh,
system.
ribosome. Okay, you're talking. Okay.
Yeah. So those 16 is rRNAs, in fact, they don't have any role with respect to expressing any enzymes, Sanjay. They are only ribosomal and they have certain unique sequences which can be used to identify the species.
Yeah. I'm. I'm not sure if ribosomal RNA. If ribosomal RNA have. Sorry, what?
Vandita, you can go on. Any other
The
questions?
the audio is it's kind of really muffled. I don't know why, but I'm so sorry if I
No,
don't.
we can hear you clearly, no problem.
Hi, Vandita.
Hi,
This is
This is
Priya, madam.
Priya.
who is this? I'm sorry.
This is Priya.
Oh,
Priya,
Priya.
ma'am.
Hi, Priya, ma'am. Hi. It's been so long. Hi,
I
how
know.
are
How are
you?
you doing? How are you
I'm good.
doing?
How are you? I was so nervous to do this. You know.
We're all so proud of you, Vandita.
Thank you so much. Thank you so much. It's because of you guys, really. Honestly.
Your hard work always. I
How's
just,
yeah?
yeah, sorry. Go ahead.
No, you go ahead. That's fine.
Yeah. So I just wanted to tell all the students, look, most of you are in final year, right? And I'm sure you have some plans of going abroad. Many of you are taking up the GRE. You can, in fact, ask Vandita what
Yeah,
can be done, which university to apply to, what is your interest. Please do not, be laid back.
yes,
Yeah, come on, everybody.
yes. So
Rashika,
there
come
are
on,
two.
Rashika. Rashika's around. She was interested in going abroad, I suppose, or Shreyas.
In fact,
Shreyas is around.
come to us, no, ma'am, PM ma'am, when we are there,
Exactly.
us and ask a lot of questions. When we have someone who has gone and achieved so much and ask them to, ask their doubts and queries, they don't ask.
Absolutely.
No, there are essentially two reasons you want to go outside the country, right? Because the lifestyle is attractive and also because the research is attractive. But I do have to tell you this, that research in India is booming as well. You know, there's so many grants that the Indian government has for people to pursue science. You know, you can do that as well. That's another alternative. If you do want to go outside, that's great too, you know. So if you have any questions about going outside, please let me know. I'm sure there's like, you can email me too if you want. I'm sure I can put it up in the chat, or is my email visible?
You can put it up in the chat box.
Yeah, so you can email me with any questions you have. You know, I'm easy to talk to. I don't bite. So, you know, feel free to email me or ask questions right now. It's a quiet class, huh?
Come on, people, you don't have any questions.
Vandita, you're currently in India, right?
Yeah,
Yeah.
I am.
Okay. How is
Uh,
the scenario out there, the US, with respect to funding and funding opportunities? I mean,
so
it doesn't, I've heard it differs from state to state, right? So how is it in Texas?
Texas, the funding is really hard to get. You know, the first time my PI got funded for his work, you know, he was overjoyed. It's a very selective process, but the funding is tough and it's very
It's
competitive out there. It's very competitive. Because
right.
they have the best institutions and you're competing with people from, I don't know, all the Ivy League universities that work nonstop, you know.
Oh, yeah, I know.
So it's just,
Must be facing a very stiff competition from them,
yeah,
though.
and their funding is directly tied to their political scenario, which is,
Yeah.
yeah, which is absolutely garbage right now.
Exactly. I guess it swings between the Democrats and the Republicans, I suppose.
Yeah, it's unfortunate, but that's
Very
how it
unfortunate.
works there. So the you know, under the last administration, funding from NIH was actually cut.
Yeah.
And now I'm not sure what this administration is going to do. Anyway, it's a sticky situation. I don't know about that.
Yeah, everybody has some hope on funding. I hope
Yeah.
the NIH also opens up their eyes and start the funding process.
Yeah, you know, in India, I do hope that they sort of tap into all the universities that we have for research. I don't know why we aren't doing that. Only select universities do that, right? IITs, NCBS, IISc. We have so many young students. They're all so brilliant, but you know, our government doesn't tap into it, all the potential that we have. Hopefully that changes.
Exactly.
But there's some amazing work going on in IITs. I was talking to somebody, a professor there, and it was really great. Um, so I'm here for like a year, and I'm leaving. I was talking to Rashmi ma'am about this. So I'm hoping that I can move to and work under him in one of the IITs, and then leave. You know, it'll be
Okay.
awesome.
Oh, that's nice. So
Yeah.
when do you plan to, get back to the US? What is your next plan? How about a PDF position? How easy is it to get one?
So I actually had a postdoc position in UCLA, but I turned it down because of personal reasons, and I decided to take a break and come back to India. I hadn't seen my parents for about 4 years or something, and then now I'm gonna spend a year here with them, and then I'm going to Canada. Actually, my brother is essentially Canadian, So we decided as a family to, set up shop in Canada. So I'm going to be looking at postdoc positions there in Canada. I have a few in mind, but I'm going to apply for those later in the year.
That's nice. I mean, Canada is also, a lot of opportunities. Small place, not a small place though, but in terms of its population.
Yeah. Obviously,
So the
America.
competition is also, I guess, less than what you see in the United States.
Yeah, it's obviously America's gold standard for some research activities, but I want to balance, my work with the family. I have family in Canada, so I want to be close to them. And PhD is not easy. It's very, if anybody who wants to go outside and who's interested in doing a PhD, it is not easy. And it might be a cliche that I'm saying this, but it really is not. It's a lot of hard work. It's a lot of no sleep nights, and you just have to put in a lot of effort. So you have to talk to people who've done it, and make your decision before you pursue anything. So yeah, so that's why I wanted to, hopefully after my postdoc there, I'll transition into like industry. That's my idea. I definitely don't want to be a PI. That's my personal decision. But yeah, postdoc for sure.
Very nice. Wishing you all the best.
Thank you. I definitely couldn't teach. I've seen you, ma'am, and you guys. You're way better at it than me. I'm not a good teacher, so
You are, Vandita. Whatever you taught, we understood it very clearly, right?
thank
So, teaching comes with, research experience only. So you have done so much of research, and obviously you can be a really good teacher also.
Absolutely.
thank you so much. It was so nice. It was so nice talking with you. And if anybody wants to email me, please do. I'm really friendly. I have like that nostalgia about MVIT, so I'll help you.
Thank you, ma'am.
Anybody from the final years and pre-final years? Come on, people, what's stopping you from saying a hi at least?
They're scared. They're scared of me. Come on.
I'm sure they will have, thousands of questions, but I don't know, they just don't want to ask it.
Yeah, no, that's okay. If they want to email me, that's fine too. I understand stage fear.
But there's no stage here. Nobody can see them. And no one knows, you don't know any of them. And this
Yeah,
platform is for them actually, it's not for us. We always, feel happy and interact with you people and get to know things, it's fine. But they are the ones who have to, reach whatever heights that you have reached, or they have to follow the paths that you have followed.
thank you so much. But is anybody who's interested in, doing fundamental science or getting a PhD, is anybody hell bent on doing that in the 81 people who are here, or the students?
Okay, hello.
Hi. Hi,
Okay,
Alan.
yeah, hi, I'm Alan, and I'm planning on doing my further studies in the same field you did it in,
Okay,
molecular
that's
and cell
great.
bio.
Oh,
So,
that's awesome.
yeah,
Have you started applying?
I haven't started applying, but I'm thinking of Europe as a potential place.
Yeah, they have really good opportunities too. Germany and Sweden. I'm not sure about what the immigration process is like in Europe if you eventually want to stay there, but I do know they're doing some great work. I have some friends in Sweden, so that's great. Good luck with that.
Thank you.
Hello.
Hi, Dhruv.
Hi, how are ya?
I'm good. How are you?
Yeah, just fine, hanging in there. I actually had a question about how do you get the unrestricted right to live and work in the United States? Or how do you apply? Any idea?
Unrestricted?
A
You
residence,
mean how do you become a citizen?
a residence permit initially. I know getting a citizenship is harder.
Uh, yeah, it is, but well, with respect to PhD, it's not. It's actually an easier route to get citizenship. I just chose Canada over USA, but it is hard. You know, it's tied to the political situation, and because of COVID, right now they will not consider any of that. Do you know what I mean?
Yeah, right. But uh, I was asking with respect to any job in general after finishing every graduation possible.
Oh, yeah. So, if you finish your master's there, say in, I don't know, business or something, and you do well and you get a great job, your company loves you, they'll give you your H-1B visa, and you know you'll get your green card. But you know you have to be good at your job and keep it. And it's a long process, you know, it's not like 4 or 5 years. It takes like 10 to 12 years for you to finally become a citizen. So they're really selective about it.
Okay, cool. Thanks a lot.
But if you do a PhD and you get a nice publication and you get a certain number of citations on your publication, you know, they'll be like, okay, we'll fast track your permanent residence or whatever. So maybe you could do PhD.
Also,
Right.
I guess I'm just adding on, I suppose you need to write some exams with respect to American history, right? To, in fact, that's the first step to, applying for a citizenship itself.
Yeah, but that's way down the line.
Yeah, that's, I think, one of the earliest steps.
Yeah,
So you need to
first,
clear that to even, go ahead in your application, I suppose.
yeah.
Ma'am,
I
that's
mean,
mostly for the United Kingdom.
what?
Even the US. Even the US, you need to write an exam. You need to take a couple of exams on the American history.
Yeah,
The first thing you need to do is memorize all the names of all the presidents.
okay. Yeah, that's true.
That's
But
going to be very hard for us students, yeah.
I mean, to get a citizenship, they'll do anything. They'll memorize anything.
Yeah, that's also true.
They won't even know our president, but they'll definitely know
Correct.
who was the 15th president of the United States.
yeah, I understand your sentiment and the need, the interest in moving out, but it is tough. Right now, we're unlucky because of COVID as well, and you why don't you focus on your career more than the goal being to move. Why don't you focus on something that you're interested in and then apply and then do well and citizenship will follow,
That's a very good point.
right? So MS in biology, there I'm not sure about MS in biology, but you could apply for MS in biotechnology in universities. That's good. Also, another advice: if you are applying for any of these fields, research fields, why don't you try and do an internship here? Because NCBS and IISc, they do take interns during the summer, and they if you work well with the PI, it'll be amazing on your CV, and it'll be easier for you to transition into their education culture, their academic culture. So I think, if I don't know if anybody here is interested, try and intern with somebody who's actively doing research here. you know Tell them that you have classes, you'll only do work later, it's like a part-time thing, but I suggest that you do that. Be resourceful, be active in looking for these things, because they do matter. I interned in NCBS, actually, 1 year into my master's, when I came back for a holiday, and it was great. It was it was the reason I got into Dr. Campbell's lab, because I had this experience. So I suggest that you do that.
Hi, I had a question.
Hi,
Yeah,
Priya.
yeah, hi. Actually, when you mentioned about working in the industry after your postdoc, did it mean you want to work in the R&D department of a company, or what is it?
Yeah, R&D department. There are industry postdoc jobs that you can apply to. And they're more professional. It's not academia. You're not going to be teaching and things like that. Also, it doesn't have to be wet lab work. You can apply to industry positions where you're an editor, or you're reviewing something, or you're a counselor. So there are a lot of things that you can do in the industry. But I do want to stay in the research field for a while right now, at least, because that's what I like right now.
Okay, so it isn't hardcore research work, right? If you go for an industry?
It can be, if you choose, if you want it to be, but it doesn't have to be either.
But it'll be like a salary-based job, right?
What?
Will it be like a salary-based job?
Salary based?
Like
Yeah,
a monthly salary?
yeah. Postdoc is a
Okay.
job too. Postdoc, you get money every month too, but
Okay, okay. All
just
right.
in academia.
Yeah, yeah. Thank you.
Thank you. Good luck.
Any more doubts? Any more questions from students?
I had one doubt, ma'am.
Yeah. Hi, Sanjay.
Ma'am, how is the people's view on biotechnology in Canada and other countries, ma'am?
How are people's views on biotechnology and?
On in other countries, ma'am? Will they accept
Uh,
biotechnology, or it is like just like in India, ma'am?
so I'll answer the question that I understood was asked, but so biotechnology is booming, right? It's tied to biological sciences. So we usually, all the labs in biotechnology and in basic biology departments, they collaborate. For instance, I collaborated with a lab in biomedical sciences, and we published a yeast paper on shape-changing material with yeast in them. So, it is a good field. And they're actively funded as well. They're funded well. This particular lab, this biomedical lab, had military funding, and, it was great working with them. So it's good.
Thank you, ma'am.
Any more questions?
Hi, Vandita. I joined late. Sorry.
Oh, hi, Dr. Nagendra.
So nice to see you after a long time. Wonderful.
Yeah,
Thanks
yeah,
for
it's so nice
connecting.
to talk to you.
Yeah, yeah. Thanks for connecting. So suddenly an unscheduled meeting came up at 10:30, so I had to miss your first half of the session, maybe. Yeah.
No, don't worry about it. It's absolutely fine.
Okay.
Yeah,
Yeah,
I'm
great.
happy. I'm happy to be involved in any way.
Okay, thank you so much. So nice to read your profile on LinkedIn and then get to know that you have a gained rich experience. In fact, wonderful. No,
Thank you.
very nice publications also. I just browsed through those publications as well. Very nice.
Thank you so much.
Excellent.
Yeah, hopefully.
So you have finished your doctoral thesis or is on cards?
Yeah, I finished it, but I couldn't walk, because of
Okay.
just when I was about to graduate, the whole COVID thing happened. So
Okay.
they canceled all the grad walks and no hooding ceremonies. So
Okay.
it
Okay.
just
Okay.
mailed me my degree certificate. It was sad.
Okay. Kindly share that certificate with us. I think it will be a valuable document for our files as well.
Yeah. Should I email that to
Yeah,
you?
please.
Oh,
A soft
okay.
copy of the same. Please email.
Yeah,
Okay.
I'll do that.
So
But
very
I
nice. So you are Dr. Vndita, but congratulations.
Oh my God. Thank you so much.
Yeah, it means a lot. It means a lot. I know. I
Yeah,
know the struggle. Yes, very important.
it means a lot that, my undergrad professors are proud of me.
So kind of you. Okay, we deeply appreciate your gratitude.
Thank you. So I was telling all the students if they had any doubts or if they wanted to talk to me, just talk to me. That's fine too. They can email me. I'll help in any way possible.
Excellent. So what is the plan? You are currently in Bangalore. That's the understanding.
Yeah. So currently I'm staying here for about a year. I was telling Priya ma'am and Rashmi ma'am, and I'm talking to a professor who has a lab in Madras.
Okay.
So I'm trying to get a postdoc position there for a
Okay.
year, and then I'm moving back to North America.
Okay.
So that's the. I went about it in an untraditional way. Traditionally, they just do postdoc there right away. But
Correct.
I decided to turn. I had a position. I decided to turn that down for some personal reasons, and come back home and see my parents before going
Good.
back.
Very good. Very good.
Yeah.
And when things become better, I think you should also visit and then maybe talk to students
Yeah.
on some more scientific work that you have done.
Yeah. I was hoping. I could do that sometime. In
Yeah.
the near future.
Yes, maybe January, February, whenever it's possible. Kindly visit the department.
Yeah, whenever you are open, I'm more than happy to come see you all. But the vaccine might not be out till
Yeah.
mid-year
Yes.
next year. So I don't know. I don't know what's going to happen.
But how about your Madras assignment? Will it be offline or online then?
Oh, it'll be online.
Okay.
So his lab and my lab, we used to be in communication, when I was doing my PhD. So we worked on similar things.
Okay.
So he's more, willing to take me in
Excellent.
because he knows my work. He knew my PI and stuff like
Very
that.
good. Very good. Very nice.
Yeah.
Great. I think we will request Rashmi to formally thank. Maybe Jagdish has a question. Yeah, Jagdish.
Hi, Vandita.
Hi, Dr. Jagdish.
Yeah, fine. How are you?
How are you?
Yeah, fine. Yeah, I was in the class, in this cell
Yeah.
biology class. I joined late. I could not, even I could not attend your talk. So
No,
15
that's
minutes back,
okay.
I joined. Thanks. As Professor said, I was also going through this thing. I saw your message in LinkedIn, but late.
Yeah. No, that's okay. I understand. You're active, like you have so many classes to teach. That's fine. It's so nice to talk to you, though.
Yeah, me too here. So good to see your growth. So
Yeah.
you're all the inspiration for all these juniors. We kept, telling about you all. I remember the day you left for US.
Yeah.
In the last.
Thank you so much for everything.
Yeah.
I wish I hope all the restrictions are lifted and that I can come see you and talk to you personally.
Yes.
But yeah.
No, I'll be keep showing all of our alumni's LinkedIn photos in the classrooms.
Yeah,
So I've shown
there,
many
there are
times.
so many. There are so many from our batch who are doing so well.
Yeah.
Right. So I heard Tejaswani is in UK, right?
Yeah.
Yeah.
Vanagi Millen.
Yeah, and somebody else. I forgot, but that's we were a good batch.
Yes.
We did okay.
Yeah. It's a great thing. Thanks for talking to students. Okay.
No, thank
And
you
you're
for
all inspiring, indirectly to all juniors. Because as I said, like in my classroom, when they get bored, I'll open up LinkedIn. I'll keep showing all your, whenever I see some of your LinkedIn, that means I'll be showing to the junior. Yeah.
Yeah, thank you so much.
Thanks. Okay.
Bye. Have a good one.
Yeah. Bye.
So if we don't have any more questions from students, can we all join hands and say thank you to Vandita?
Thank you, Vandita. Thank you so much.
Thank you, everyone.
Thank you, ma'am.
Thanks so much for listening to me
Thank
on
you.
a Saturday.
Thanks, Vandita, and hope to see
Thank
you
you.
soon over here.
Yeah, me too.
Yeah.
Good luck with everything, guys.
Thank you.
Thank you. Bye bye. So
Bye, Vandita.
should I end the call?
Yeah, thank you so much.
All
We
right.
hope to see you continued association with you.
Yeah, me too.
Thank
Thank you.
you.
Bye.
Bye bye. Bye bye.